Excitation contraction first occurs when the neuromuscular junction uses the nervous system to send a signal, which is the stimulus for the muscle to start its excitation contraction sequence. The stimulus or signal sent by the nervous system is also known as ACH, acetylcholine. When action potential reaches the axon terminal, calcium floods the neurons, resulting in communication between the stimulated neuron and targeted muscle cells. The muscle cell membranes are called plasmalemma. This connection resulting in the communication is called the neuromuscular junction site. This site is where the motor neurons transmit its signal to the muscle fibers, initiating muscle contraction. The muscle contraction is initiated by the depolarization of the sarcolemma caused by the sodium ions associating with the acetylcholine receptors. Immediately after this depolarization, it reestablishes polarization, allowing for the action potential to travel along the T tubules so that it can reach its destination at the sarcoplasmic reticulum, or the SR. The T tubulars are arranged with the sarcoplasmic reticulum on the sides, also known as a triad. The triad surrounds a cylindrical structure called myofibril. A protein called DHP receptor senses the membrane potential on the T tubules and then relays this information to the RYR receptor on the SR membrane. The RYR receptor provides a channel for rapid calcium release from the SR, and the DHP receptor acts as the trigger for the release of calcium from the SR. Once the action potential sweeps along the sarcolemma, it releases the calcium ions that interact with shielding protons known as troponin and tropomyosin. These shielding proteins are moved out of the way, allowing for troponin, which acts as a switch, allowing the muscle contraction to occur. Tropomyosin keeps actin from sliding between the myosin, hence the shielding. The myofibril is the location where actin and myosin are contained. ATP attaches to myosin, giving it the energy to start the contraction. The myosin is a thick protein that pulls on the thinner actin, causing the actin to slide over the myosin, creating a muscle contraction. After the muscle contraction, the sarcoendoplasmic reticulum calcium ATPase, or SERCA, acts as a pump that transports the calcium ions from the cytosol back to the SR. This allows for the reuptake of calcium